Hello and welcome to day 8. Today I'm going to talk about the bacteriophage, which is the biological entity with the most number on Earth. Unlike its name, the bacteriophage um, attacks bacteria and is actually a virus. So let's delve deeper into these small monsters. So basic knowledge you need to know about the bacteriophage is that they are not actually bacteria, they are a type of virus right here. And the name bacteriophage comes from the word bacteria and phage, which means to eat. So literally it means to eat bacteria. And as I mentioned before, they are the biological entity that has the most number on Earth. They are very deadly and we are beneficial that they are not hostile towards us humans. If they were, we don't know what would have happened if they were hostile to us. So the big structure of the bacteriophage consists of three big parts: the head, tail, and the tail fibers. The head is the shape of an icosahedron, which is a 20-faced, 30-edged dice. And inside the head, the genetic material, the DNA, is contained inside of the head, and the head is coated with capsid, which is a protein coat that protects the head. No, not the head, the genetic material inside the head. Next is the tail. The image we're looking at right now, this part is actually the tail itself. This is a protection layer called the chest. The tail is actually inside of the chest and extends whenever the bacteriophage attaches itself on the cell's bacteria surface. And the tail supports the head. And the most bottom part of the head, of the tail right here, is also known as the base plate, the end of the chest, as you can call it. Fiber tails, no, yeah, tail fibers. Tail fibers help the phage attach to target bacteria. They connect to the bacteria's receptors, and yeah, they are basically for connecting to the bacteria surface, and we're going to learn about that as we go on. More features about the bacteriophage. It is very interesting. They only kill a specific kind of bacteria and its close relative. So the bacteriophage has had a lot of species themselves, like bacteria, which have like so much kinds we can't even count them. Yeah, there are a lot of other kinds of bacteriophages too, and they each have a specific kind of bacteria to kill. And since they are a kind of virus, they require a host to reproduce, as they cannot reproduce themselves because they are neither an organism nor a non-organism. It's really obscure. They only kill bacteria and don't kill any other organisms like animals or other somatic cells inside our body. And here comes the important part, the process of bacteria destruction. This is how the bacteriophage destructs the bacteria that they are targeted to destroy. So I divided this up to 6 stages. Um, Connection, injection, biosynthesis, endolysin, lysis, and repetition. In the connection stage, the bacteriophage connects its fiber legs to the bacteria's receptors, and they use a sort of syringe that come out of the base plate to right here to puncture the surface of the bacteria. And in the injection stage, the phage squeezes its tail through the punctured area, so the tail comes out uh, of like inside of the chest and extends through the punctured area of the bacteria, and it injects its genetic information into the bacteria. And ultimately, the bacteria gets taken over by the phage, and they are forced to reproduce new phages. And how this works is bacteria have um, um, a copy machine to reproduce and copy their genetic material and components to reproduce themselves. However, if the phage connects and inject their genetic info into the bacteria's copy machine, the bacteria are forced to reproduce the components and genetic material of the bacteriophage. Um, and this keeps on going, producing until the bacteria is filled up with new phages. And one detail right here is the genetic material are formed after the protein code is formed because they need to be protected and not um, harmed while being produced. If that happens, the proper bacteriophage, will not perform its function. And once the bacteria is filled up with the phages, the phages emit an enzyme called indolacin. And yeah, it is an enzyme that punches a hole in the bacteria, which makes the pressure increase. And this leads to the lysis process. In the lysis process, because the pressure increases so much, the bacteria emits all of its insides and dies. Yeah, it's kind of gross, but by this stage, the insides mean the filled phages that are fresh and yeah, freshly reproduced. And the new phages are released, and they repeat the cycle again over and over, and that's how they kill bacteria. Yeah, imagine that happened to us humans. It would be really painful and harmful. So, since we learned about the characteristics of the bacteriophage, let's go a bit beyond this. Since humans have discovered penicillin and antibiotics, they have created numerous kinds of medicine and antibiotics that killed many kinds of bacteria. And however, once we thought we had eradicated all the bad bacteria, they have went through countless evolutions and developed a resistance against our precious and useful antibiotics. And therefore, we have a term now called a superbug. And this superbug is a bacteria that has resistance to all human medicine, all of it. And quite cases are actually happening, like quite fatalities are increasing due to these resistant superbugs. But there actually is a way to go against these superbugs and kill them. And it is not a human medicine, it is actually a natural killer called the bacteriophage. So, as I mentioned, the superbugs go through evolution and advancements to develop resistance against our medicine, right? The bacteriophage does the same, but in a different mechanism. As the superbugs evolve, the bacteriophage also find a way to um, find the weaknesses of the superbugs and attack them in order to kill them, as they are, um, and as this is their sole purpose of living. So to use an analogy, it's kind of like a predator and prey mechanism. Because prey has to get away from the predator, which is basically stronger. They develop fast pace and poison to defend themselves. However, predators also have developed new traits to catch the prey since they have to survive too, like sharp fangs and claws and poison, like develop poison themselves. So yeah, so we compare the bacteria to the prey and bacteriophage to the predator. That actually quite makes sense, right? Yeah, so there's actually a case where an incurable disease caused by bacteria in the chest. Uh, I don't like quite remember the name of it, but uh, it was incurable by all antibiotics and um, remedies that humans have developed. However, once doctors injected a few thousand of the bacteriophage directly into his chest where the bacteria lived, within a few months the bacteria have completely disappeared and had been eradicated by the bacteriophage. And although this seems like a good idea, it's still in development, as we don't know if it's really safe for us humans. However, huge scale uh, research are going on nowadays to convert the convert these killers into our helpers in order to eradicate all the bacteria living in our world, the harmful bacteria. And also, fun fact: the antibiotics also kill the beneficial bacteria to us, since they're beneficial bacteria to us, not only harmful ones. However, the bacteriophage only gets to the specific harmful virus they are programmed to uh, exterminate. Yeah, and that's really interesting, and can make a great tool for us to terminate all the harmful bacteria that um, um, makes like the body susceptible to them, resistible. So basically, that is all for today, and I will come back with with more interesting content. Thank you for listening.